Good afternoon, everyone, and good afternoon, ma'am. Praised be Jesus and Mary. This presentation will be revolving around the intestinal cystole parasite of cats and dogs, or also known as the Dipylidium caninum. The objectives of this lecture that should be achieved at the end of this presentation are: first, is to comprehensively discuss the information that is connected to this parasite. Next, is to know the interaction and the relationship between these parasites and how it affects us humans. Lastly, is to enhance our knowledge and expand more of it through the additional information that I have included in this presentation that will be given as we go along. Now, let's start. This parasite is known as Dipylidium caninum, pronounced as Dipylidium caninum, which has many common names such as cucumber seed tapeworm, pumpkin seed tapeworm, flea tapeworm, and double port tapeworm. And as we go along this lecture, we will be able to know the reason behind this parasite having many common names. To start with, this parasite or the Dipylidium caninum is the most common intestinal parasite of cats and dogs worldwide, which needs a flea as an intermediate host for it to complete its life cycle. Therefore, its range is dependent upon the availability of the flea and the vertebrate host, as well as its capacity to survive outside the host until it is ingested again by the larval flea. It occurs mostly in dogs where ectoparasitism is high. Ectoparasitism or ectoparasites are the type of parasites that live in or on the individual's skin but not within the body. Examples are the fleas and the lice. This parasite is a monoecious and a hermaphroditic parasite. Both of them are slightly similar, but when we say that the parasite is monoecious, it has the both male and the female reproductive organs on the same species, but it is just located on different parts. When we say it is a hermaphroditic, it has a gender ambiguous reproductive organs, both containing the male and female reproductive organs on the same individual or species. Next, it is responsible for the disease dipylidiasis. The transmission is through the ingestion of infected flea containing cysticercoid that can be transmitted by hugging or kissing our pets infected with the flea. The history of this parasite started on 1758 when Linnaeus recognized and named this parasite as Tinea canina or Tinea canina. On 1863, Leucard created the genus Dipylidium. On 1893, Diamare described the parasite, and on 1936, Neveu Lemaire reported the early life cycle of the parasite. Epidemiology: it is cosmopolitan in distribution, which means that there are cases that have been reported worldwide. Human infection is rare but have been reported in certain countries such as in European countries, in the USA, in the Rhodesia, China, and in the Philippines. The habitat is in temperate, tropical, and terrestrial regions. For the morphological stages of this parasite, on the left portion of this presentation is the one I've made. I've made a table which summarizes all the appearances, all the sizes, all the shape, and all the other information that is connected to the morphological stages of the parasite, such as the egg, the proglotids, and the adult worm. This is to help us compare and contrast the parasite and somehow visualize it in its different morphological stages. To start with, the morphological form of this parasite is the egg form. The parameters included is the diameter range per egg is 30 to 60 micrometer. There are 5 to 30 eggs per packet, and the shape is spherical. Other features include that these eggs have a thin shell with hexacanth embryo. When we say that it is a hexacanth embryo, it is the embryo that is found on the tapeworms under the subclass Cestoda, which has hooks, and these hooks allow them to attach themselves to the gut of the vertebrate host. In Dipylidium caninum's case, it has 6 hooks located on the posterior end. Notice that the eggs are closely packed together. This is because the egg packets are formed after the sexual reproduction, and the uterine lining encapsulates the egg. On the lower right portion is the image of an egg of the Dipylidium caninum that can be viewed, or that is viewed under the microscope. Next is the Dipylidium caninum's proglotid form. Proglotid is one set of reproductive organs. There are three types of proglotid forms in the Dipylidium caninum: the immature, the mature, and the gravid proglotid. When we say that it is an immature proglotid, it means that the organs, or the reproductive organs, are not yet well developed. It is the opposite of the mature proglotid because the reproductive organs are now well developed and can be seen. For the gravid proglotids, the reproductive organs is not that obvious anymore because the inside walls are now filled with eggs that are pumpkin seed in shape. For the better picture of the Dipylidium caninum's mature proglotid is here, which is composed of the genital pores, the vagina, and the vagina is just below the genital pores, the ovaries, the ootype, and the testes. For the better picture of the gravid proglotid is here, the uterus is enlarged and the inside walls again are filled with eggs. Since this parasite is a hermaphroditic parasite, it does not need any more another adult worm for it to reproduce because it only needs itself. Then, the mature and the gravid proglotids are the same because they are narrower and broad. However, because of the egg capsules, the walls of the gravid proglotid are deformed and the uterus are enlarged. For to compare and contrast the mature from the gravid, the shape of the mature proglotid is not yet well identified, whereas the gravid proglotid's shape is pumpkin seed, thus the name pumpkin seed tapeworm. The appearance is This is the tricky part of the Dipylidium caninum because this is the reason why the parasite is known as the double-pored tapeworm because of the appearance of the mature proglotid, which is narrow with two sets of male and female reproductive organs with bilateral genital pores. Then the gravid's appearance is it is full of capsules or packets of about 8 to 15 eggs enclosed in an embryonic membrane. Next morphological form is the Dipylidium caninum's adult form. Here is the picture of the scolex or the head of the Dipylidium caninum. The scolex or the head bears the suckers and the stellum, which allows this parasite to attach itself to the bowel wall of the vertebrate host. Then the size of this adult form of the parasite can range from 10 to 70 centimeter, but other references would say that it can grow longer until 10 to 50 centimeter. The number of suckers are 4 and their stellum is present, as I have said earlier, club-shaped, armed with 1 to 7 rows of hooklets. This is the picture of the Dipylidium caninum's adult form. And another picture that can be seen with its proglotids clearly, and then another zoom in picture or better picture of the proglotids when seen in the feces. Before I proceed to the discussion of the life cycle of this parasite, allow me to introduce first the intermediate host, which is composed of the Cynocephalidis canis or the dog flea, the Cynocephalidis felis or the cat flea, Pulex irritans or the human flea, and the Trichodectes canis or the dog louse. The difference between the flea and the louse is that the flea jumps from one host to another while the lice only crawls. Also, even without the host, the flea can survive up to 100 days, unlike the lice that dies after 2 to 3 days without the host. The accidental host are the humans, especially the children under 8 years of age and also the infants, since this stage is their hand exploration stage, wherein they just hold onto anything without knowing if it is harmful or not, they still do not know about the proper hand washing. Likely, transmission could have occurred by accidental swallowing of the arthropod host through hugging or kissing their infected pet. Infective cysticercoid is the infective stage, and the proglottids are the diagnostic stage. Again, since we are only the accidental host, let us first discuss the life cycle of this parasite in the animals. As I have mentioned earlier, in order for this parasite to complete its life cycle, it needs the flea as an intermediate host. It starts its life cycle as an egg which lives in the infected host and excreted as feces. Each proglottid contains egg packets that are held together by an outer embryonic membrane. The proglottids disintegrate and release the egg packets. Free eggs are rare. The most common is the package eggs, which have the mechanism that allow them to find their way out on their own. Outside the environment, the flea larvae comes across with these eggs. Since it eats anything it can find, such as an organic debris, it ingests any tissue or fecal debris that the Dipylidium caninum parasite is stuck into. The ingested egg hatches and transforms itself into an oncosphere that has 6 hooklets. This oncosphere attaches itself in the hemocoel or the larval body of the flea, and then it develops into a cysticercoid, which is the infective stage of the parasite. Over time, the larval flea molts twice, and then it transforms itself into an adult flea. The cysticercoid remains in the flea as it matures from a larva into an adult. The vertebrate host becomes infected by ingesting the adult flea containing the cysticercoid. The cysticercoid breaks free and starts transforming itself into an adult flea in the small intestine of the vertebrate host. And since it has hooks, it attaches itself into the bowel wall and begins growing body out of its neck. The body is composed of independent segments called the proglottids. Each segment has both male and female reproductive organs, which is why they are said to be hermaphroditic parasite. Since it has no digestive tract, the adult worm absorbs nutrients through its surface from the gut contents of the host. As the parasites get longer and longer, and may reach up to 70 centimeter long, it is difficult for them to pass through. Thus, it starts letting go of the mature and the oldest segments. Eventually, the segment outside the environment dries out and breaks into parts, gets consumed again by any nearby larval flea, and the cycle continues. And since we are only the accidental host, humans can be infected, especially young ones, by the ingestion of the cysticercoid contaminated flea by the close contact with flea-infested pets. The scolex attaches in the intestine, and the adult in the small intestine develops. The gravid proglottids again are passed intact in the feces or emerge from the perianal region of either animal or human host, and the cycle continues. The disease associated with this parasite is dipylidiasis, which is proven to be a zoonotic disease. Zoonotic disease means that the dipylidiasis have been transmitted from animals to humans. Infection is rarely heavy and symptoms are minimal. Patients are also asymptomatic. The symptoms are related to the worm burden. However, if the worm burden increases, the symptoms may also intensify. The patient may experience intestinal disturbances such as slight intestinal discomfort and epigastric pain, diarrhea, and anal pruritus, and allergic reactions, intestinal obstruction with severe mechanical occlusion of the lumen, which only happens rarely in indigestion, and also appetite loss. These are the symptoms for you to know that the dog or the cat is infected with such parasite. This is important for pet owners for you to monitor your pets. Weight loss may occur. Sometimes the dog will scoot or drag its anus across the ground or the carpet due to severe itching, and the proglottids can sometimes be seen crawling near the anus or on the surface of a fresh bowel movement. The Proglottids that can be seen in the feces excreted by your pets resemble the rice seed, the pumpkin seed, and the cucumber seed. Hence, the common names pumpkin seed tapeworm, cucumber seed tapeworm, rice seed tapeworm, and flea tapeworm. The laboratory diagnosis includes first is through the recovery of gravid proglottids in the stool sample, either single or in chain. Proglottids should be pressed or flattened between two slides. Stool sample for the presence of eggs is not recommended since the proglottids do not disintegrate in the intestine but rather on the environment. Egg capsules are rarely recovered from the stool. The treatment of choice if you are infected with such parasite is the praziquantel 5 to 10 milligrams per kilogram given on a single dose and is taken orally. For the prevention and control, the best way to prevent the dipylidiasis infection, especially for children, are to control the fleas that infests your pets. Flea collars, medications, and powders can help in reducing the flea of your pets, and also periodic deworming of pet cats and dogs are also advised. Insecticide dusting of pets against the fleas. Do not allow children to play in areas that are soiled with pet or other animal feces. Also, teach children to always wash their hands after playing with dogs and cats and after playing outdoors. Here are the nice to know facts of Dipylidium caninum. The segments contain autonomous muscle fibers that allow them to move on their own, enough for them to exit from the perianal region of the host or from the stool sample that they have been released with. The proglottids are narrow with two sets of both female and male reproductive organs and bilateral genital pores, thus gaining the common name double-pored tapeworm. The proglottids also resemble rice seed, cucumber seed, and pumpkin seed. Hence, the common names of this parasite. Adults are not commonly infected with this parasite due to age tolerance, and the first human infection was reported in 1912 by M.P. Mendoza in a child in the Philippines.